Hello and welcome back. In this video we're going to introduce the key online bioinformatics resource providers, the NCBI and the EBI. Now this is the home page for NCBI at the time of recording. Now if this website, when you visit it at this URL here, looks slightly different than what's shown here in the slide, it's because websites change and morph their appearance over time. Now typically, major content does not disappear, but you may have to click in a slightly different place to find some of the things that we're going to link to in the next video and in this one. So just be persistent and I'm sure you'll figure out how to navigate these resources. Now you've probably used the NCBI or the National Centre for Biotechnology information before. For example, in this popular resources over here in the sidebar, it lists PubMed as number 1 here. So this is a database of biological or biomedical literature that you may have come across previously. But they also host popular tools including BLAST, that's for sequence-based searching of sequence databases. If you haven't heard of BLAST before, and some of you will of course, we're going to hear much more about it in a subsequent video. We'll figure out how BLAST works and its underlying algorithms, and we'll apply it extensively. There's also listed here databases for nucleotide, for genome, for SNP, that's single nucleotide polymorphism data or variant data, for genes, for proteins, and then this last one, PubChem, here this is for small molecules or chemical entities, typically, most typically things that are drug-like, that interact with biomolecules in important ways that we often want to know about for biomedical research. So the most notable NCBI databases for the purposes of our course, the ones that will interact in these initial portions of the course, include GenBank and its derived database RefSeq. This is for nucleotide sequences. We're going to interact a lot with those in the Handson session and indeed throughout the coming weeks. We'll also use the search tools Entrées. This is for typically text-based searching across all of NCBI, across all their different databases and resources. And then we'll also, as I've already said, we'll use BLAST, this sequence search tool. Now over to our second major bioinformatics resource provider, that's the EBI here. You'll notice that its URL is based out of the United Kingdom. That's the .uk at the end here. The EBI, it stands for the European Bioinformatics Institute. It's actually based out of just outside Cambridge in the UK. It maintains a number of very high quality curated bioinformatics databases and a lot of associated tools. Also, I'll point out in the second red box there, they actually have a lot of training material. That's video resources. Well, you could see in the background of this slide, for example, that'll walk you through and introduce you to using a number of the major EBI resources and bioinformatics tools. They can be particularly useful. Now, for the purposes of our course, we're going to deal a lot with UniProt. This is a protein sequence database, one of the premier databases out there. It's actually a model for how we wish many bioinformatics databases were organized and arranged and maintained. We'll also interact a lot with Ensemble. This is for genome information. And we'll use the tools, in particular Muscle. This is for multiple sequence alignment. And also Hammer, which is a hidden Markov model-based approach for searching sequence databases, much in the same vein as BLAST, but with a number of important advantages and also a couple of limitations that we'll touch on as well. It's meant to be more sensitive. In fact, it is more sensitive, but it can be dramatically more time-consuming, as we'll see. Now, there are many good, notable bioinformatics databases out there, of which I'm only listing a small but still scary subset on this slide here. Now, quite frankly, it would be crazy and really rather boring actually to start to go through any of these in any sort of detail at this stage of the course. But instead, what I want to point you to is a carefully annotated listing, a PDF file that I have on our class website that annotates these major databases by particular application area. So the purpose here is, if you end up in the future in a fly genetics lab, or you join an immunology group or a company, or you end up in a plant lab, or you work in a structural biology group, you'll be able to go to this handout and find the most widely, best regarded, most highly cited databases for each of those particular biomedical research areas. So this will be a useful resource for you in the future, rather than to try and dig into any of these other non-NCBI and EBI resources at this particular time. Now, as you go through maybe exploring that PDF, there's an important side note. That is that not all databases are created equally. And just like these cakes here, some of which I made with my kids here during this Corona lockdown, for example, right? They contain the same ingredients as some of these cakes. They were meant to look like this. And this is the case for many of these databases. They were meant to, or indeed they do contain the same biomolecular entities as some of the other databases, but they're not always going to be as good to interact with. Just like these cakes, they're not necessarily the ones you want to eat or select to eat from the ones that are available. So what we'll do is we'll explore what makes databases good for our particular application areas as we go through in our course, and we'll use many of these resources to help answer various questions and see what makes a database good, what makes it valuable, that it has the information, of course, that'll help us answer our biological questions, but also that it's able to be searched in efficient, tractable and meaningful ways, and link out to other good resources that we can learn more from as we go through. So this hands-on session that I mentioned, you'll find it on our class website. So at this stage, what I'd like you to do is to go back to our class website. That's thegrantlab.org, BIM 143. We're in week 2 currently as I record this video. So click on the schedule, go to week 2, and you'll find a link there to lab. That's our hands-on session. What we're going to do is we're going to explore a number of these major databases, primarily from NCBI and EBI, but also some others, as we investigate what's going on with a particular patient that's come into the clinic with some unknown condition. We'll get a sequence, as we'll see, and we're going to go through like a detective and try and explore what could be wrong or what could be going on, and actually get a molecular understanding of the basis of this patient's condition. So the hands-on sheet, when you click on it, it'll look like this. For this week, it's a PDF. It has fields in it and questions in it that you can fill in and save the PDF file with your answers. The major areas, there's a number of different sections, 4 major sections that I want every student to go through. The first one goes through these NCBI resources. It'll probably take you 20 minutes, but I'm allocating around 35 here. Then the gene database at NCBI. Then I suggest we take a break for some time and then come back and we'll jump over to Europe, to the EBI, and we'll visit UniProt, the premier protein sequence database. We'll use Muscle, an online tool out of the EBI web servers there, as well as some of these boutique databases, PFAM, which is also maintained out of the EBI, but the PDB for protein structure data, and this NGL, this is for viewing biomolecular structures in an online browser. Now there are a number of questions throughout those sections. I do want you to everybody please answer the last question in that workbook. That's a kind of what we call a money point assessment question. It asks you, what is particularly unclear from what we've covered this week, and it lets me know what maybe we're not doing a good job of getting across, and we'll try and record another video or talk in the forums or in our Zoom office hours and get those points across in a different way. We do encourage discussion online in the forums and exploration. So please do, if you find something interesting as you're going through the lab sheet, don't be scared to click off and go follow it up. Exploration and focus on something that you actually are interested in are going to help you learn this material a lot more clearly than just following a recipe that I give you, for example. So with that, I'll finish up this video with a summary of what we've talked about this far, namely that bioinformatics is computer-aided biology, that bioinformatics deals with the collection, archiving, organization, and interpretation of a wide range of biological data. It's not tied to any particular method or any particular subfield of biology, but it's as wide as biology itself. We've introduced the NCBI and the EBI. These are 2 major online bioinformatics service providers. And in the next video, we're going to actually walk through the gene, UniProt, PDB databases, as well as a number of other resources, including PFAM, protein family database, and the OMAN database. This is for human disease conditions, and they're linked to the genetic basis of those diseases. So we'll see you next time. Thank you.